Okay, so this is a lecture on muscular and musculoskeletal systems. It's not going to be very long, but I'm going to be very repetitious when I say a lot of things, and I might add some elements to make the concept of contraction more comprehensive. Because with regard to musculoskeletal system, a lot of the questions on the MCAT, from my experience or practice tests, deal with the mechanism of the contraction and deal with the concept of the levels, the release of calcium, at when it's used, at what type of muscle it's used for, and then whether certain muscles show certain characteristics, whether certain types of muscles are mononucleated or like have many mitochondria. So we'll talk about those, and we're going to also talk about some aspects like the bone and small parts of it. You don't really need to know an extensive amount about the bone, but you do need to know like slight details about it and how it's structured and how it's composed of. Like the difference between osteoblasts and osteoclasts, what's an osteoid, stuff like that. So I wrote all these notes out, and I'll eventually put it on Pre-Med Star. I just need to organize all my notes, because when I write them down on OneNote and make them to a PDF, they look a lot larger, and it kind of like comes out of the page. So I have to like arrange them so in order for you guys to see it, because I write them all out on OneNote. So I have to figure out. But once I do that, I'll post them all on Pre-Med Star. But I have posted from my glycolysis lectures and I think my lac operon lecture, I posted a couple documents. You can look on the note share button on Pre-Med Star website on the left hand side, and you can look up my name and you'll find the documents I uploaded. There are also documents that other Pre-Med Star members have posted, and which are really helpful to help you study for the MCAT and to just do study skills, and I highly encourage that you go on there. And if you have anything that you find useful, or that you've made for studying and you'd like to share with your fellow pre-meds, go ahead and post it. Like all of us are on the same road, and like if you want someone to help you out as much as and like if you like everyone should treat each other like should try and help each other out. So you want someone to help you out and give you tips. It should be, maybe can be vice versa. As we're all really a community, and we should all try to like keep working together. So that's why I try and make these lectures very interactive and very helpful. So here we go. So if you want more notes, you can go there. And I think I think a lecture was posted earlier today regarding, I think it was my electrostatics lecture already. So you can look at that if you want to rewatch it. And this will be posted in the next few days. Okay, the musculoskeletal systems. So generally, the functions of the muscle is body movement, stabilization of the body position, and this kind of also is associated with the brain. And when I do my lecture eventually on the brain parts, the parts of the brain, you'll know that this is a very important element when dealing with muscles. I think, or from the top of my head, I think it's the basal ganglia that deals with the stabilization of the position. And another element that deals with like stabilizing your body position, or orientation rather, is the fluids in your ear. They help you orient yourself so you know what space you're in. So that's the point of fluids. They have other functions to them, but I'm just adding them to kind of connect for you guys. The functions of the muscle is also involved in the movement of substances throughout the body. So you want to think of muscles as kind of being the machine for the brain. So the brain wants certain things done, but it can't do it on its own. So the muscles are basically the machines that move stuff throughout the body. The bones are controlled essentially by the muscles. The muscles orient the bones to move to move around via the nerves, neuromuscular junctions in the muscles and stuff like that. So movement of substances throughout the body. In cardiac muscle, it propels blood throughout the body. Smooth muscle helps regulate blood flow and uses peristalsis to move food and waste throughout the body. Peristalsis is essentially when your food's going down your esophagus, it's the muscular movement. Well, not muscular movement, no, I'm sorry. It's like basically the process, the automatic process, like the involuntary process of food, and etc. moving down esophagus. And you know this because, like, you don't have to, I mean, maybe sometimes you have to force yourself to swallow something, but, like, most of the time the food you eat comes down by itself, and that's through the process of peristalsis, which the smooth muscle helps be done. That wasn't very articulate, but oh well. Let's go. I'm just putting this here. So if you guys have any questions, just let me know. Out loud or on the chat. I don't mind if you interrupt me. I'm here to help. Okay, so the first type of muscle is a very important one, is dealing with skeletal muscle, and I'm going to be talking mostly about this today. So with skeletal muscle, it deals with the voluntary movement of somatic intervention. So basically, voluntary. It basically means the muscle you use that you manipulate voluntarily. So the muscles in your fingers, the muscles in your arm, the muscles on your legs, like stuff that you voluntarily move that aren't automatic. And it can only be manipulated or mostly manipulated by your intervention or your brain's intervention. In the skeletal muscle, the cells are arranged in fibers, and I'll go into the orientation and the specificities, like the specifics of how these fibers are arranged. And the cells in skeletal muscle are multinucleated. This is a very important thing to remember. It differentiates it from what's it called? Cardiac and smooth muscle. So I'm gonna highlight this. Because you see in a picture, when you have a picture of a muscle cell, I can't come off the top of my head what it looks like. So you'll have, so it's multinucleated, and because of that, it'll have striations. So also skeletal muscles have striations, which are basically stripes in the muscles. When you get it, if you do histology, you basically take slides of like tissue and striations. If you take a muscle cell and put it on maybe you did this in like your bio lab class where they showed you different types of muscle tissue, and you would notice when you look inside the microscope, you see these stripes and multiple dark red dots or dark dots, because in histology, when you take a slide of tissue, after you do that slide of tissue, you have to stain it. That's why you see color whenever you get a slide of tissue. It's not, I mean, maybe sometimes it's natural, but most of the time it's not. It's the staining, so you could differentiate between the two different types of tissues. So in this case, if you were to take a slide of skeletal muscle, you would see basically stripes and then a bunch of darker dots, and those darker dots would be the multiple nuclei, and then the stripes would be the striations, and the striations are due to the main part of the skeletal muscle, which is the sarcomere. And the sarcomere is a contractile functional unit of the muscle cell, and they're very important basically because skeletal muscles are basically utilized all the time. They need to be automatically used and always contract. Need readily contraction. It's not going to be going through rhythmic motion like you would for a cardiac muscle, because in the cardiac muscle, it's controlled by the brain, right? Or rather, the brain stem. And because if you guys have already reviewed your psych. Your psych terms, or the body part, or the brain parts, or you took a psych class or a neuro class, you'll know that the brainstem is where all the survival elements are for maintaining the body, right? So of course you would have the center that's controlled, you would have in that area that controls the heartbeat. So with cardiac muscle, you need to have very rhythmic movement or contractions. When you think contraction, think movement. Remember when we talked about inhalation yesterday? With inhalation, every time inhalation occurs, the lungs expand, and with that, the muscles, the external intercostal muscles, and the what's it called? The diaphragm contract in order to modify to deal with the fact that the lungs are expanding because they're taking in air. And then once because the lungs are elastic, they kind of flap back, or they like flap back, and then the muscles relax through a passive process. That's exhalation. So I'm giving that as a parallel. So with the sarcomere, it's another example of contraction, but with the skeletal muscle, it uses sarcomeres, and sarcomeres are these little elements or functional units of contraction. So those are the striped region, or they're responsible for the striped regions inside of a skeletal muscle. And they're arranged in a very regular manner, and it gives the skeletal muscle a striated appearance. I'm going to get a pink color so I can highlight that. Sorry. I like to use a lot of colors when I write my notes and I talk about things, because it makes it seem like it kind of like, for me at least, I don't know if you guys study like this, but for me, like giving different colors to it, like kind of keeps me entertained and keeps my attention open, so that like if I'm tired or if I'm bored, at least I have something to distract myself with, or if at least have like to keep my brain something to look at while I'm studying. So stimulates my brain cells, if you want to like think about that. And what's unique about the skeletal muscle is that each cell, you know how in the cell you have things like the endoplasmic reticulum and the cytoplasm and etc. Well, in a muscle cell, you would kind of rearrange, you would call them different names because there's sarcomeres in them. So in a muscle cell, the endoplasmic reticulum, or in a skeletal muscle cell, the endoplasmic reticulum rather is called the sarcoplasmic reticulum. And it basically is the same thing, but it's just located in a skeletal muscle cell. And the cytoplasm in a skeletal muscle cell would be a sarcoplasm. And so whenever you hear those words, you know you're dealing with a skeletal muscle cell, and so things are a little unique in there. And the sarcoplasmic reticulum, it's a very unique area where it is utilized to store calcium to the release during contraction. So when contraction occurs, and I'll go over the details on how that occurs, but when contraction happens, you need, in order for it to occur, a release of calcium needs to happen. So always remember this. If there's no calcium around, there is no muscle contraction. If there is no movement, you know there's no calcium being released, or rather, you know that calcium is being blocked. So like whenever you have those injections that kind of like inhibit muscle movement, sometimes they use calcium inhibitors to block calcium in order to stop the contraction. So always think this: when a skeletal muscle is moving, if you block calcium, you block the contraction and its movement. And with muscles, any movement is a contraction. If you want to stop a muscle movement using another muscle, it's another form of contraction. It's just one contraction blocks out another contraction. That's how it works. Like there's muscles like if you want to like stop another contraction using another muscle, that other muscle has to contract. Like that's essentially the point I'm saying. I'm only making this point is because I'm remembering an MCAT passage a long time ago that I took, and it tried to trick me in that, and I'm like, and that's why I'm reiterating to you. So what was I saying? So a sarcoplasmic reticulum, so it's another way of saying the ER, but it's in a skeletal muscle, and the skeletal muscle has sarcomeres, so that's why it's called a sarcoplasmic reticulum. Sarcoplasm is the cytoplasm of a muscle cell, and sarcolemma is the cell membrane of a muscle cell. So sarcolemma is another way of saying cell membrane of a skeletal muscle. Does that all make sense so far? Okay, good. All right, I'll continue. Okay, red fibers. So there are like three different types of fibers. There's red, there's intermediate, and there's white. Intermediate is just the middle of the two. I'm just gonna talk about red and white because intermediate is just going into advanced tissue function that like you don't cover until like grad school, or rather, I'm covering grad school, so you don't really need to know about that. So red fibers are slow twitch fibers. So whenever you think of red fibers, think slow. I'm gonna try and give a different color. So slow twitch fibers with increased number of mitochondria and increased myoglobin content. And that makes sense because they're red, right? They're red. That means there's a lot of energy going through. So they have a lot of mitochondria and they have a lot of myoglobin. And the idea of myoglobin, if you don't know what that is similar to hemoglobin, but only has one subunit to store to store oxygen for release when it's needed during cellular respiration. So myoglobin is basically it's not hemoglobin, but it's just a form. It's kind of like hemoglobin, but it has only one subunit to store O2. So if there's increased myoglobin, that means there's more oxygen, so it's more red, right? Because remember what I told you yesterday, if you had a hypoxia in your skin looks bluish. That's why when you see people who've been like suffocated and stuff like that, you see their skin going blue because those areas have deoxygenation, right? So if they're red fibers, it makes sense that they have increased myoglobin because then there's more O2 around. And red fibers also have a lot of mitochondria because having a lot of mitochondria means they can utilize cellular respiration, or rather oxidative phosphorylation, and utilize all that oxygen to make more and more and more ATP. Because remember, in aerobic respiration, you want to use oxidative phosphorylation to make as much ATP as possible because it is the most energy generating mechanism. Anaerobic processes like glycolysis, they make ATP, but they're not very efficient. So if you have oxygen, you want to go to oxidative phosphorylation because you're going to make as many ATP as possible. Able to perform aerobic respiration for long periods of time and used during sustained vigorous activity, long distance running. So whenever you think of long distance running, think red fibers. Okay, white fibers are fast twitch fibers that have decreased number of mitochondria and low myoglobin content. So basically it's the opposite. It's white, it has less red, less oxygenation, so low myoglobin content and lower number of mitochondria, less cells needed to utilize the oxygen. And it goes through anaerobic respiration, so it has an increased rate of contraction, but you can fatigue very fast, so like sprinting. So when you go through anaerobic respiration, there's a lot of release of lactate. Because if any of you remember, or if you remember my lecture on glycolysis, when you're going through anaerobic respiration, you don't have oxygen around. So after the pyruvate is made, okay, I'm just gonna go over it again real quick. After the pyruvate is made, and you have to memorize the structure of pyruvate. I'm not kidding you, you have to memorize it. So you have the structure of pyruvate. You remember after glycolysis, you have 2-pyruvate, right? So because you don't have any oxygen, you want to have more glycolysis occur, right? But how do you do that? So you would change pyruvate to lactate, but why would we do that? Well, we want to change pyruvate to lactate because of we want to generate more of NAD+. So we have a lot of NADH made from glycolysis, right? We want to turn it to NAD+ because it is an ingredient for glycolysis. So we want to make more of this so that we can add it on to make more glycolysis occur and change more and break down more glucose molecules. So pyruvate changes to lactate. And this is a little trick I think I said in my lecture, but I'll say it again. So pyruvate to lactate is a reduction, and this is oxidation, right? Because the hydrogen is lost, so it's oxidation. So the reduction is being powered by the oxidation. whenever you have a reaction like this reaction is going to be the opposite of this. So if you know the pyruvate changes to lactate, then you might not need to memorize this at all. You'll just know from opposites. Like if this is a reduction, then you know this is an oxidation reaction. That's how it occurs. And in muscle cells, or in white fibers, since they don't have a lot of mitochondria, so they don't have a lot of mechanisms needed to utilize oxidative phosphorylation, they're going to keep using anaerobic respiration, so keep going through glycolysis. So you have an excess release of lactate in those muscles, white muscle fibers. So when you have sprinting going on, you have a lot of a lot of lactate being released because of a lot of fatigue. You keep going through inefficient energy. You're not making as much ATP as you can through aerobic respiration, but you have it. But you fatigue very fast because that energy runs out real quickly. So that's the basic concept of it. What else is there? So now I'm going to talk about smooth muscles. Smooth muscls are involuntary. They are controlled by automatic neurons. They're mononucleated, so they have only one nucleus. When you analyze them in a histology slide. They're non-striated, but they still contain actin and myosin. So even though like there's no striations, actin and myosin are always there. Actin and myosin are the mechanisms for muscles contracting in general. It's the striations are only there if there are sarcomeres, and sarcomeres are mostly found in skeletal muscles. So all muscles have actin and myosin. Actin and myosin are essential for muscles to contract. It's just the presence of a sarcomere that's the differentiation. Or the sarcomere is like basically an arrangement. And the thing is, in skeletal muscles, they have a slightly different arrangement of things going on in there, because they want to. They're always used. So they're kind of designed in a way for constant, unique, inflexible manipulation. Contractions are slower but maintain longer in smooth muscles. They have a very rhythmic, automatic stimulation. They have myogenic activity, a reflexing contraction independent of the nervous intervention. So reflex can be associated with smooth muscles, and they don't have Ttubules, troponin, or tropomyosin. And I'll get into what those are when I talk about contraction, but these are very important elements with regard to skeletal muscle contraction. So remember these names: troponin, Ttubules, and tropomyosin. Okay, next is cardiac muscle, and cardiac muscle is pretty simple. The reason why I made it pink, because it's in the heart. It's striated, so it's like a little bit like skeletal muscle, but it has like a fewer nuclei than it would a so think of it kind of like cardiac muscle, or muscle is kind of in the middle of between skeletal muscle and smooth muscles. Cardiac muscles are striated because they're manipulated a lot more than smooth muscle, because you're always like, your heart's always beating, but they also have more nuclei than smooth muscle because of that, but it's not as much as skeletal muscles because they have a very rhythmic and very, kind of constant like movement. Automatic nervous system modulates the cardiac activity, but it also has a myogenic activity. The heart will continue to beat if all nervous connections are severed, but it will, I'm sorry, 1 second. Okay, sorry. But it will just not be able to increase or decrease the rate in response to exercise and rest. A cool example of this would be, it was like, I think last week in my lecture, and because we're doing animal husbandry in my graduate course, and we were dissecting a rat, and one of the rat that I was dissecting, after we had opened him up, he was already dead. After we had opened him up, we looked inside. His heart was, I think mine and a couple people in my class, their heart was still twitching, even though it was dead. So even though like the nervous intervention is severed, it still has the automatic like movement like it's still like beats slightly. Like it's really interesting. You can look it up on YouTube when you see dissections. So it'll twitch sometimes. So that's an example of the cardiac muscle like having an automatic, you know, sense of like beating all the time. So that's just a cool example you can think of when you think of cardiac muscles. And eventually, like I think some programs you can see, or if you ever do dissections of like rodents or basically of animals, you will notice that. So that's a cool example that I brought up for you guys. A pacemaker cells. So these are basically automatic rhythmic action potentials and contractions. I'll get more into this when I talk about the circulatory system, because it's in the heart. It has a very unique nervous or a unique like spread of like action potential, including the pacemaker network or from the pacemaker fibrils. But I'll get into that when I talk about the cardiac system. But you just know that these are there for making rhythmic action. And I think mostly in the brain, it's associated with the brainstem. Okay, so muscle contraction. I'm going to try and explain this because you do need to know this absolutely for the MCAT. So, initiation. So, when you have a skeletal muscle, or rather, this is muscles in general, but I'm gonna use the skeletal muscle as an example. Okay, so a muscle cell mostly gets stimulated by a motor neuron, which means it'll take information from the brain, or rather directions from the brain, and act it, right? So it'll be stimulated by a motor neuron and at the neuromuscular junction. And what is the neuromuscular junction? It's basically the connection between nerve and muscle. So it's like the machine, or rather the like computer telling the mechanical arm to move, in a sense, right? So at that point. So a motor neuron inside this, the connection between the nerve and the muscle, moves to tell the muscle from the nerve, oh, you have to do this. The brain tells you to do this. Go and do it, like something like that. And then a chemical called acetylcholine. If you guys did your hormone review, I mean, I'll do a lecture on hormones eventually, I think eventually, but you should know what this is. Acetylcholine is released, and then it binds to receptors on the sarcolemma. And do you remember what sarcolemma is? Sarcolemma is basically another way of saying cell membrane for a skeletal muscle. So acetylcholine comes in after the muscle got the command from the nervous system, right? It'll move and bind to receptors on the cell membrane on the skeletal muscle, and that's called the sarcolemma. And remember, if you reviewed your nervous system, like if enough action potentials or summation occurs, and it's an all-nothing process, so enough are bound, and action potential will be potentiated. So like if enough are added on to the dendrites right here, and then the cell body says, okay, you can put an action potential, it'll shoot out an action potential. So if it all adds up, that means an action potential is released. That's the beginning, right? So when the action potential is released, so basically initiation is, you can think of the initiation as like making action potential. And contraction is reacting process. So process reacting to action potential. AP, I'll just say AP. And then, okay, so we'll start with this. After the action potential is released, it enters these things called Ttubules. And what are Ttubules? Ttubules are basically whole, or wait, well, then my textbook said they were called invaginations, so, but I'd rather say holes, I don't know. It just made more sense. There are holes in the sarcolemma that allow electrical impulse to travel into the fiber, the fibril. Because with muscles, like, they kind of are divided like this. There are things called, a bundle of muscles called fascicula, I think it says, or something like that. It's a bundle of muscle, and then it goes into the muscle cell, and then the myofibril. And so the Ttubules are holes in the cell membrane of the skeletal muscle that allow the electrical impulse to travel into the fiber and cause the sarcoplasmic reticulum to release calcium for contraction. So the Ttubules, they're those little, like, they're basically like channels allowing for electrical impulse to move around and go and move around to the cell membrane and signal the sarcoplasmic reticulum to release calcium. And remember what I told you? Once calcium is released, it is essential for having a contraction occur. So once calcium is released, what happens? So like I said, I'm going to repeat what I just said. Action potential, after it was made, it enters the Ttubules. The Ttubules are the channels. It'll move the electrical impulse along the cell membrane or the sarcolemma, and it'll trigger the sarcoplasmic reticulum to release the calcium stored inside. And then the calcium released is utilized to bind to something called troponin. And I'm going to explain what troponin is before I go any further. So normally when a muscle is relaxed, troponin binds to something called tropomyosin. And tropomyosin, I'm sorry, no, what am I saying? Oh, God, that's not true. Okay, no. Scratch that, sorry. Tropomyosin is, when it's relaxed, it's blocking actin from binding to myosin, right? Or, you know what I'm saying? Because it'll bind, so tropomyosin on resting state is basically the security guard for myosin, right? It'll stop actin from binding to the myosin and stopping contraction from occurring. And so once calcium binds to troponin, it'll change the conformation, give a conformational change to troponin. So troponin will be like, I don't know how to say this, more like the ice cream truck for the security guard. And so the calcium helps the troponin transform into an ice cream truck. And so the ice cream truck goes on, and the security guard the tropomyosin sees the transformed troponin bound to the calcium, and then it moves away from its guarding of the myosin. So now the actin could go in and bind to the myosin. And so since the tropomyosin has been moved away from the myosin by troponin calcium complex, myosin head binds to the actin and pulls on it to cause a contraction. And then when a contraction occurs, or contraction of the sarcomere occurs, there are these zones. And I'll go into what these zones are basically. So let me find the picture I have over here. Okay. So in a relaxed state, there's the I zone, the A zone, and the Z zone, right? And then the H zone. So when a contraction occurs, the A zone will always stay the same. It just always stays the same. And when a contraction occurs, the Z line is usually like over here, right? And every time a contraction occurs, it'll get closer and closer and closer to the middle, right? And then with regards to the I zone just gets shorter and shorter and shorter. So like when you were contracting, it's like squeezing something. It gets shorter and shorter and shorter, right? And the A zone is the center, so it stays the same always. And the same thing with the H zone happens to the I zone. So the I zone, the H zone, like gets smaller and smaller and smaller as a contraction occurs. So when you think of contraction, A zone stays the same, the Z zone gets closer to the center, or the Z line gets closer to the center. The I zone and the I zone and the H zone all get shorter. So just remember that. Okay, yeah, the I zone and the H zone reduce in size as the Z line comes together. That's essentially, I'll just rewrite it for you, or rather I'll highlight it. That's a crappy highlighter. Okay. So next we have relaxation. So in order for relaxation to occur, you need to get rid of that calcium. That calcium is the reason why everything started, right? You want to stop it from, you want to get rid of the ice cream truck so the security guard could go back to its place, right? So rigor mortis occurs when no ATP is left to move the calcium back into the sarcoplasm. So rigor mortis basically happens when all right, I'm getting ahead of myself. So basically, in order to get rid of the calcium, you need ATP. ATP is when there's no ATP, constant contraction occurs, and that makes you have really stiff muscles, right? And that's why when someone is dead, basically, you find the muscles in the body become very stiff. It's because there's no ATP to stop the contraction of the muscles, so the muscles will stay stiff. And yeah, that's basically with that. Neurotransmitters are released to diffuse across the synaptic cleft and attach to the acetylcholine receptors on the sarcolemma. So this basically repeats what I was saying. The action potential generated is propagated to the along the sarcolemma and down the T-tubules. The action potential triggers the calcium release from the terminal cisternae of the sarcoplasm reticulum. So basically, action potential triggers the calcium to be released. The calcium ions bind to troponin, and troponin changes shape and changes the shape of tropomyosin and stops tropomyosin from blocking myosin from actin. Now actin and myosin can bind, or rather the actin binding sites on the myosin are exposed, so now actin can bind to myosin, and contraction occurs. And during contraction, the Z lines get closer and closer to the A zone. The A zone stays the same. The I zone and the H zone get smaller. Contraction, myosin crosses bridges, alternatively attached to actin and detach. So the myosin pulls the actin filaments towards the center, basically squeezing it forward. That's why you see the I zone, the H zone gets smaller and smaller as contraction occurs. The sarcomere, then there is a release of energy by ATP hydrolysis powering the process. So when a contraction occurs, ATP is broken down, basically. So in order to drive like the power stroke, basically, of the pulling and the squeezing of the actin and the myosin. Okay, and if you guys remember what active transport is basically use energy to drive a movement of a concentration against this gradient. So a primary active transport is when this is done via ATP, and this is an example of primary active transport. Secondary active transport is basically the same concept, but you don't use ATP, you use another form of energy. The removal of calcium. So after the power stroke occurs, you want to stop it, so you put in ATP, and that ATP or active transport powers the movement of the calcium back into the sarcoplasmic reticulum. So I'll write that down on the side. So you want to get rid of calcium, but to do so would be energetically unfavorable. Or rather, remember yesterday what the term was I used for this? Do you remember? Yes, but what's the other word? Yes, that's exactly, yeah, that's actually, you know what, actually, that's right, but another way you could say is non-spontaneous. You said the right word, though. This is just another way of saying it, non-spontaneous. So good job, you did know that word. It's endergonic. Another way of saying it's endergonic, because it needs an input of energy. But that, you're right, though. That's absolutely right. Yep, exactly, a delta G positive. Good job. See, you're learning. That's good. So you want to get rid of the calcium, but to do so will be an energetically unfavorable, non-spontaneous reaction. So you need an input of energy, right? So this movement of calcium away against this gradient is primary active transport, because we use ATP. Move calcium back into sarcoplasmic reticulum after AP ends via primary active transport. So we use ATP. ATP has come back. ATP used. And so when ATP is around, you know contraction ends. So whenever you think ATP, contraction is ending. And if there's no ATP around, think about like a dead person, basically. A dead person has very stiff muscles because now they're dead. They can't produce any more energy. They don't have enough ATP. They don't have ATP in their bodies to relax those muscles. So that's basically the understanding. And where was I? The tropomyosin blockage, restored blocking of actin, binding sites on the myosin, contraction ends, and the muscle fiber goes back to its original position. Does all that contraction concept make sense? Okay, good. All right, good thing. All right. Next, I'll talk about the skeletal system. So joints. Joints are basically, there are like three types of joints. You don't have to know like the specifics, but you should know like the basic understanding of how they're separated. Joints, basically, there are three types. There's fibrous joints. So basically, the bones are held together by fibrous tissues, and it's immobile. And so an important element to know about this one is that it's immobile. I'll erase this and rewrite it. An example of a very big example of this is the skull, because you think about it, it's very immobile, right? You can't move it around at all like an arm or something. I think so, actually, yeah. I do think it does go back in pumps, but I'd have to double check. But yeah, because like in order for calcium, you had use an ion channel technically, like because it's a ion, well, you know, calcium 2+, right? But it needs to be triggered. So they're basically like They're not like the types of ion channels you'd find in the nervous system, right? That are like that determine the resting potential, right? No, like the calcium pumps that are on the sarcoplasmic reticulum need to be initiated by something by something binding to the receptor and then releasing it. So yeah, I think it is pumps, but I'd have to get back on you on that. It doesn't, but it works basically. Something has to bind the acetylcholine or has to initiate action potential. Action potential and it goes and instigates the sarcolemma to release chemical, and the chemical binds to, and that's all cell bio. Um, uh, it's a basic in cell bio. I think it has to do mostly with the G protein coupled receptors and changes configuration. It releases a certain modified phosphate group, and it releases when it cuts down the molecule. That cut down molecule goes on and binds to the receptor on the sarcoplasmic reticulum, and then the sarcoplasmic reticulum releases the calcium through ion channels, and that so no, it doesn't work via pumps like it does in the nervous system, but it goes through ion channels that are triggered by a specific molecule binding to the receptor on the sarcoplasmic reticulum, and it works as a secondary cascade, basically. It's an example secondary cascade. Does that make sense? Okay, good. I'm like remembering it from the top of my head at the moment, so I'm gonna look it up later and then send a doc on the note share, because that's actually a cell bio concept that I think it's in the chapter in the Kaplan book. From the top of my head, what chapter was it? It's the one covering G protein coupled receptors. Go on the chapter that talks about G protein coupled receptors and see the section that talks about cyclic AMP, and that will tell you about how it triggers the release of calcium from the sarcoplasmic reticulum. I didn't go over it in detail because that's mostly to do with cell bio, and it's beyond the it's kind of like everything in MCAT sciences, and the sciences is connected, but I didn't want to confuse anyone by bringing it up. But that's an example of the cell bio, the secondary what was it called? Oh my god, I'm spacing out right now. Cascade. This is the example secondary cascade. The cascade that's responsible for making sure there's a release from calcium from the sarcoplasmic reticulum when initiation occurs. So that's basically the idea. No problem. So what was that? Okay. So next, the second type is cartilaginous joint. Bones are connected by cartilage and relatively immovable. So ribs, sternum. Synovial joints are bones separated by fluid-filled capsule, which has a large range of motion. So like your knee, fingers, and stuff like that. An example of this is like, I think when I was in high school, when I was in my first year in undergrad, I fell down the stairs, and then I had hit my knee really badly, and I could feel that there was fluid that had inflamed that had like gotten out and inflamed my knee, and so I couldn't move for a while. And my father, he's a doctor, so he was telling me how I had to like basically let it heal on its own for a while. And so in a sense, like with your knee, if you the synovial fluid is there to works as a lubricant to reduce tension basically on your knee. So that's an example. My injury is an example for you to remember what a synovial joint is basically. So there you go. We all have injury stories that help you. Oh no, yeah, it's better now. It just took a while for it to heal back in the day. So don't run down the stairs on a train platform in the city. Just don't do it. No, it's not worth it. So what was I saying? Okay, the ligament is basically connects bone to bone. So bones can't touch basically because it causes a lot of tension, and it causes a lot of problems as well, and you'll feel a lot of pain when it does happen. So you need ligaments in order to make sure that doesn't occur, and tendons basically connect the muscles to the bone. Skeleton, there are two ways you can see the skeleton: the axial way, which is where the skull, the vertebral column, and the rib cage are, and the appendicular, where the limbs and pelvic bones are. So basically think the center of the body is like the axial, the appendicular is basically like your arms and legs, like the appendages. Okay, cartilage, chondroitin sulfate and collagen matrix. So it's a mix of these two chemicals, which are both released by things called chondrocytes. Cartilage is basically made by these cells called chondrocytes. And the only reason why I know a lot about this is because in my grad lab course, we did a lot of like experiments where we had to break down cartilage, because cartilage is a very tough substance. You can't break it down by just proteolytic enzymes. You have to strip away the cells from the tough tissue and then try and break down the elements inside cartilage. So in the lab, it's a very tough substance to try and break down. You can't just use pure enzyme to eat it all away. External ear, you find cartilage basically in external ear, nose, walls of the larynx, trachea, and skeletal joints. There are several types of cartilage around. But there are three types that you just need to know about. The Highline, which is the most common, it's found in joints and the ends of long bones. And then the ends of long bones is important to know. They have these, there's these esophageal, I think they're called esophageal discs. I might be wrong, but I have to remember the name. But they're basically like holes in your bones that stay open while you're growing, so it releases all these chemicals and stuff like that. And they have to close at some point so that you stop growing. And they close around the time when you're an adult, when you reach maturity, so around like 20 or 18 years old, depending on if male or female. And like dwarfism and gigantism, if you know the actor from Princess Bride who played the giant, he was actually a famous wrestler in his own right, and he had a condition where his the seals in those ends of his long bones didn't close until very late until very late, and so he kept growing and growing and growing, and it was basically the reason why he could basically drink a lot. My professor talked about this, how he would drink basically 2 cases of beer and be completely fine because he was so huge. So my professor brought this up, not me, and I thought it was a good way for people to remember. So that's one example, and dwarfism is when the seals close too quickly, I believe, from the top of my head. Yeah, so, so that's an example you can think of, gigantism. He's probably the most famous known person, though he's not around anymore. He died, I think, in 1997, I believe. I don't remember, from like a heart attack. I have to, it was you can watch The Princess Bride, you can see like how his condition it looks, or how his condition basically is an example of like the long bones, and how if that area doesn't close at a certain point in your maturity. And then fibrocartilage is another type of cartilage, which is found in intervertebral discs. And then another type is elastic. So just know that there are 3 types, and these are the 3 types. You don't really need to go into detail what they are. Okay, the bone. So there's compact bone, which is very dense. There's spongy bone, which is I'm not exactly sure. I think it's mostly found in your legs and arms, but I'd have to double check. I don't know exactly. I haven't covered many the details on cartilage yet. Sorry. Okay, so spongy bone creates medullary cavity inside large bones. So when you see the picture of the inside of a bone, you see like the spongy end of it. That's the spongy bone. The trabecular is the bony specules that creates a bony network inside the compact bone. So it's basically a network within the bone so that it can like grow. So like bone remodeling is basically when you change immature bone cells to mature. So that's where this comes into play. Cavity between the trabecular have yellow or red marrow. Yellow marrow is basically where inactive infiltrated fat tissue or adipose tissue occur. And then red marrow is the importance. So it's responsible for blood cell formation. So an interesting thing to remember is that with with people who have blood cancer, right? They go through these processes where you want to go through a transplant, like a bone marrow transplant. Red marrow is where you want to target. So we had, I think, in one of my exams, we had a question where we had to look at an initial scan of the genetic makeup of the bone marrow that was being removed, and then the genetic makeup of the bone marrow that was going to be transplanted, and then the makeup of after the patient had gone through the transplant. And you'll see in every single type of transplant, you'll find that there's like a slight leftover of the previous bone marrow, even when you transplant the new, because like when you find this new curve, the area that had the previous genetic makeup of the old bone marrow should be gone, and the new one from the transplant that was transplanted should be found in the patient's profile. But then, like with some patients, the reemergence of the cancer could occur, and there's like a slight leftover amount of that bone marrow genetic makeup. So that's a cool parallel for you to reflect on, because I'm remembering it from one of my exams. So like with cancer, it's a very tricky disease. So you always have to be careful of the fact that it can always come back, because nothing is very static in science. Okay, bone structure. It is made up of organic molecules like proteins, collagen, and glycoproteins. It's also made up of inorganic stuff like calcium, phosphate, and hydroxide. Calcium is important, of course, for bones, because when bones are being formed, they basically they take calcium from the bloodstream. So they use calcium to build the bone, and then when the bone needs to be broken down, it gets rid of calcium. The hydroxyapatite crystals are just mineral salts made of calcium and phosphorus, because the bone system in the body is also a form of mineral homeostasis. An osteone is basically the structural unit of a bone. You should basically know that this is basically the structural unit of a bone. So in other words, it's like the bricks in the building that is the bone. Haversian canals, that's important. Those are the areas in the bone that contains blood vessels and nerve fibers and lymph ducts, because bones themselves don't have a nervous system, or rather don't have nervous systems connecting it, nervous vessels on them, and the bones themselves don't have blood vessels. It is the Haversian canals that give the blood vessels a chance to come in and give nutrients to the bone marrow and all that. So know what a Haversian canal is. It has blood vessels and nerve fibers and lymph ducts. So basically connects the bone to the rest of the body and lets it get the materials it needs. So it's kind of like, what was it? Like the embassy for the body inside of the bone. The lacunae are spaces that contain mature bone cells, which are called osteocytes. Remember how I told you immature bone cells are woven. So, and then in order to become mature, they have to go through something called bone remodeling. And then when they go through bone remodeling, they're mature and they become osteocytes. The canaliculi are small canals for exchange of nutrients and waste. And you need to know the difference in osteoblasts and osteoclasts. Like, don't confuse them because it'll it'll come back to haunt you. So osteoblasts make bones. So don't get confused with blast. Just think when you see blast, think the opposite. Like, so it's not to trick you. So with bone, whenever you see blast, it's opposite. So osteoblasts make bones by taking calcium from the blood, and then osteoclasts break bone by releasing calcium into the blood. So it gets rid of the calcium so it can break the bones down. Types of bone formation. There's endochondral ossification. You can take existing cartilage and replace it with bone. You can use another way of making bones is long bones, growth plates in children. Remember I talked to you guys about the gigantism and dwarfism are basically like when these growth plates get closed too early or closed too late. That is essentially the case. They have to close at some point when you're growing. So, and they're found in long bones. So this is another way of bone formation. Intermembranous ossification. It has to do with mesenchymal tissue, and with regards to mesenchymal tissue, I'll talk about this eventually, maybe when I go over cell differentiation and like reproduction. So mesenchymal tissue is basically when there's undifferentiated embryonic tissue. So this is tissue whose fate hasn't been determined, right? When this tissue whose fate hasn't been determined, it is transformed and replaced by bone. So that's another way of making bone. And that is my lecture for the musculoskeletal system. Do you have any questions? Yeah, okay, I'll go that way. Yeah, no problem. 1 second. All right. So intermembranous ossification. So before I go into that, I'm going to say what a mesenchymal tissue. So when you go through when tissues are first made, right? All of them have to have their fates determined, right? Before all tissues are to be determined as kidney tissue, or to be determined as heart tissue, or to be determined as I don't know, leg tissue, whatever. They all have this form called mesenchymal tissue. So this is when it's undifferentiated. Its fate has not been like determined yet, right? Or it has not been like declared, in a sense, rather. It hasn't been changed to anything yet. So an intermembranous ossification is when this mesenchymal tissue is transformed into bone and replaced. That's basically a way of making bone. It's basically making bone from undifferentiated tissue. Does that make sense? Okay, good. Do you have any, where does it end up? What do you mean? Oh, it's the bone. It becomes the bone. So like it determines like, so instead of determining all these, it replaces like whatever it was going to be determined. And it's like, oh no, it's going to be bone. Bone is going to be made. No one else. No, don't apologize. I'm here for a reason. Do you have any other questions? Did I go too fast? Is there anything you need clarification on? Okay, that's good. Well, I'm glad it was helpful.